Okay, so my report is about protein synthesis. So, what is protein synthesis? Protein synthesis is the process by which amino acids are linearly arranged into proteins through the involvement of ribosomal RNA, transfer RNA, messenger RNA, and various enzymes. So the next slide is called the central dogma. The central dogma is the fundamental principle of molecular biology because it summarizes how the genetic information in DNA becomes expressed in the amino acid sequence in a polypeptide chain. So DNA transcript for RNA and RNA translate to protein. Okay, so in this slide, we have DNA versus RNA. So in DNA, we have double-stranded, and RNA, we have single-stranded, and in DNA, we use deoxyribose sugar, and in RNA, we use ribose sugar, and for DNA, it occurs in nucleus, and RNA occurs in cytoplasm. So when it comes to bases, DNA use thymine instead of uracil. So in RNA, we use uracil. So in protein synthesis, we have main step, which is initiation, and initiation, elongation, and termination. So in the first process, we have transcription. So transcription is the process by which DNA is copied to mRNA, which carries the information needed for protein synthesis, in which a particular segment of DNA is copied to RNA by enzyme RNA polymerase. So in the first process of transcription, um, the RNA polymerase binds to the promoter, and the DNA strand unwind or unzip, and the polymerase initiate RNA synthesis at the start point on the template strand. So that is the first step in transcription. For the second step, the polymerase moves downstream, unwinding the DNA and elongating the RNA transcription to 5' to 3' in the wake of transcription. The DNA strands reform a double helix band. And for the third step, the RNA transcript is released and the polymerase detached from the DNA. So that's all for transcription. And for the second step, we have translocation, which is the process in which cellular ribosomes create protein. In translocation, we have messenger RNA, ribosomal RNA, and transfer RNA. So, for the first step in translocation process is when an enzyme called amino acid tRNA synthesis attaches amino acid to their corresponding tRNA molecules using energy from ATP. So each amino acid has its own tRNA molecules with the anticodon for amino acid. And for the second step, we have and for the second step, a small ribosomal unit attaches itself to the 5' end of an mRNA strand. And for the third process, it moves along the mRNA until it finds a start codon. There, the first RNA and large ribosomal subunit joins. For the fourth process, other tRNAs with anticodons matching the mRNA codons fall into place in the ribosomes. For the um, fifth process, the first tRNA drops off its amino acid, breaks off, and leaves to pick up another amino acid, and the second moves over to make room for another tRNA. And for the sixth process, the tRNA after tRNA drop off their amino acids. They form acid linked by peptide bonds. And for the last process, when the ribosome reaches a stop codon, it releases the finished polypeptide. So after that, the polypeptide falls into an active protein and performs its function in the cell. And for us to identify the amino acid of codon, we have to base on the table. like this. So as you can see, the first example is AUG. First, you have to find the first letter where is A. So A. And for the second U, we have to look at the second letter U. And for the last, we have to look for the third letter is G. So the result is methionine. Same goes on the another codon. That's all.